biology is having its golden age. I mean, think about it. What has happened in physics ever since Einstein's general and specific relativity and the discovery of gravitational waves that proved certain concepts about it? What has happened in chemistry recently in the last 5-10 years that is groundbreaking, that's going to change the way we live and how we apply technology? But when you come to biology, it's just ongoing discoveries, very cool stuff, one after another. In 2006, Shinya Yamanaka figured out that we can have, cells, stem cells, from adult cells. Let me explain you what that means. Stem cells are cells that are in the very beginning developmental stages in the body. They are like the initial progenitor cells in our body, which keeps on dividing and self-renovating and differentiating into more specific cell types. So we always thought that the progression of differentiation in our cellular code goes one way, which is from more general to more specific. A zygote divides into first 3 germ layers, the endoderm, ectoderm, mesoderm, and then they divide into more progenitor cells and they differentiate into specific neuron cells. And we thought that directionality was one way. But Shinya's lab figured out that the directionality can be inversed. So you can actually take a neuron cell that's highly specialized and highly differentiated, and you can use 4 chemical factors called the Yamanaka factors to put them back into the original, like revert them back into the original progenitor cell type, which is called an induced pluripotent stem cell. This is huge. It's got the Nobel Prize and it has incredible applications already in the sciences, and also it will keep on having incredible scientific and medical applications. Right after that, in 2016, 5-10 years later, came the CRISPR-Cas9 gene editing technology, which again is world-changing, mind-blowing. It's essentially the fastest, most effective, the cheapest, and the most specific way we ever discovered to alter not only the human genome, but any kind of genome. So when you put these 2 technologies together alone and start dreaming about what can be done with combining the last, what we have figured out about biology within the last few decades, the sky is the limit, really. So think about this example. In 2016, a science research group, decided to run an experiment where they used the induced pluripotent stem cell technology to take a single cell, a skin cell from adult mice, and then use these Yamanaka factors to revert this cell back into its progenitor stage, essentially into an embryonic stem cell. Step 1. Step 2, they used the technology of CRISPR-Cas9 gene editing to add a gene in this single cell which gave it green fluorescence. It's called the green fluorescence protein, GFR, GFP, I think. And then, after doing that, they took that cell line and they started multiplying those cells to a point where they had a solid colony of cells that all share this GFP green fluorescent protein. And then what they did after is fascinating. They took half of those cells as a group and they put them under the mechanism that's needed to create a testosterone producing, a sperm producing cell, a Leydig cell. And the other group, they put them under the differentiation process so that that cell will start producing viable eggs. Guess what they did next? They put that egg and the sperm in the same cell. They basically merged the egg and the sperm, and they created a viable zygote, which they planted into another adult mice's uterus, and green little babies of mice were born. So that means, first of all, that they found a way to produce and reproduce from a single organism. There's not a mother and a father in this scenario. the entire cell populations came from a single adult mice in the first place. This means that in the species of mice, we can essentially take an adult mouse and reproduce the next generation or give viable next generation using a single adult individual, which is crazy because in biology, in general rule of thumb, if you can do something in mice, you can do the same thing to humans within the next 10 years. We catch up on the technology. Our genomes are not too different. Essentially, the same mechanisms that we apply in mouse genome apply in human genome as well. We just need to perfect the process, troubleshoot, and we might be looking into a future where a single individual might be having a baby, and not only a normal baby, but a green baby. And the analogy of the genetic editing and the CRISPR-Cas9 technology giving birth to a green mice can go a long way when it comes to altering human genetic abilities, like having stronger bones, having blue eyes, having an athletic body, having a musically talented brain connections, like neural connections. And the list is endless. In fact, there are companies in today's world whose entire job is to collect the genetic information of people who have supernatural abilities, like having strong bones that do not break, or having a sixth sense, or having an intelligence beyond the average human intelligence. And they are taking the genome sequences and storing their genetic information in the hopes that one day we'll figure out if any of these supernatural traits are linked to genetics in the first place, and who knows how we can implement the power of genetic editing, precise genetic editing using CRISPR-Cas9 technology to influence the future generations of humans and their traits, and what implications will that be in our society as a whole. Food for thought. You guys should check it out.